Luc Henry. I'm a Swiss citizen. I'm the CEO, one of the three co-founders of a company based in Switzerland called Limula. And what we do is building automation to address the manufacturing side of cell and gene therapies, and in particular, focusing on CAR T cell treatment. So the starting point of what we're doing was the complexity of CAR T cell manufacturing, and then we understood it's actually common to most of these ex vivo cell therapies. It's complex processes in an industry that never really had fit for purpose tools because they're so new, they're so innovative from a treatment perspective that there wasn't a good manufacturing process in place. So I think what we've seen is the last 5 to 10 years, an industry trying to look for solutions to address that problem of manufacturability, scalability, quality control. And we're now at a point where there are new tools coming to market. People start to build and sell fit for purpose technologies. And so it's just a matter of time for the industry to actually adopt these new tools. Now, the other observation that we make in the market is they were invented in clinical centers. Hospital clinicians were at the basis of these innovative treatments, and they've been taken over by pharma. And for a few years, it wasn't clear would pharma actually manage to produce these treatments coming from a very clinical academic original manufacturing process. And now we see the market splitting into pharma succeeding in doing this at large scale in a centralized fashion, but also a lot of hospitals taking responsibility for manufacturing in a point of care. And so it's very exciting to see these two different approaches, equally valuable. I think both will cohabit. And as a company, we're trying to serve these two markets equally well. The unique feature of this treatment modality is that the cells that you want to manipulate are the actual product down the line. And I think it was a big challenge for the industry looking at what the biotech tools industry would provide in the past, where the cells were a factory to produce a product, usually a monoclonal antibody, but the cells was not the product. And so we had to rethink completely what is a processing step? How do you manage that the cells are happy all the way through the process, that the process is kept sterile the whole way, because also the final product cannot be sterilized. It's a live cell. It's a patient cell that returns to the same patient. So I think, looking at these challenges around sterility, around the cells being the product, the new tools have to be much more gentle than they used to be. They have to take care of the cells and making sure the cells are perfectly happy and comfortable across every unit operation. But also, you need to keep that sterility. You don't have a second chance. A sterility break is the end of your batch. The last point is because they're so personalized, they usually at a very much smaller volume than usual biologics. So we had to invent and readapt technologies that they would manage small volumes, and they can be as low as 2, 3, 4, 10 milliliters, equally well as what we were designing in the past for tens of thousands of liters eventually in large scale biotech. Our unique selling proposition is in encapsulating the complexity of an end-to-end process into a single instrument. And this is something we could achieve because we made an invention where, for the first time, we combined the functionality of the bioreactor, which is what you need to incubate the cells ex vivo during the process, with the functionality of a centrifuge. And so suddenly we have this ability to process cells at the same time as we can incubate them without having to transfer them between, at minima, a bioreactor and a centrifuge between different unit operations. So that's really what's very unique about our technology, our inventive step. And we implemented this into a product we call Lim1, something that we will be launching in the next couple of years. We have an early access program open, so we're already working with a number of end users, customers, to demonstrate the value of that technology, but very much already proven that we can cover a CAR T process end to end, and we're very excited about that. Limula is currently fundraising a Series A. We're looking for 20 millions to continue our journey and take our current fully functional prototypes, turn them into products, and start commercial activities. So this is very much what we're looking for at Bio is strategic partners, investors that would support us in that next step of the journey of growth. We are invited by the Swiss government to join what is called the Swiss Pavilion. So if you're at Bio, come see all the incredible companies and not just Limula, but all the good companies that come from Switzerland and that will be exhibiting in that context.